Hello and welcome back. The topic for this video, we're going to talk a little bit about the advantages of sexual reproduction versus asexual reproduction. And that main advantage of sexual reproduction is we get variation, okay? Now remember, when we generate those gametes from meiosis-1 and meiosis-2, the 4 cells at the end of meiosis-2 are not all genetically identical. They're going to be different. Okay? Whereas with mitosis, which is going to be a type of asexual reproduction, the goal of mitosis is to generate cells that are genetically identical. Okay? So the parent and offspring are going to have the same chromosomes from the same origin, all right? With meiosis, you're going to have some differences that are going to happen. And we're going to talk about the 3 types of genetic variation that can be generated with sexual reproduction, okay? The first one is going to be that you've got random fertilization. So when you have a mating pair, all right, whether you're talking a human mating pair or if you're talking about a different species, the egg and sperm that meet up are going to be random. They're going to be randomly selected, and they're going to have a random assortment of chromosomes in them that come from that particular individual, okay? So the random fertilization of the egg and sperm, that's the first way that we get variation. This is why, for example, if you have siblings, you may not look exactly like your siblings because it was a different egg and a different sperm that created you versus your sibling, okay? Unless you happen to be an identical twin, and then the chromosomes that your identical twin have and you have are the same, okay? Fraternal twins are different. Fraternal twins would be more akin to siblings, all right? But identical twins are kind of an exception, all right? So you've got random fertilization there. That leads to variation because you don't look exactly like your siblings, okay? The next one is going to be the concept of independent assortment. And we haven't talked about independent assortment yet. We're going to talk about it a little bit more when we get to the chapter on genetics itself, okay? But what we've got with independent assortment essentially is going to get to how the chromosomes line up on the metaphase plate, okay? And what we're talking about here would be, it could be either metaphase 1 or 2, but I'm going to go with metaphase 1 for this, just out of simplicity's sake, okay? So in terms of our chromosomes, I'll draw them as big and as small. Put that over there, okay? And then we've got little one here. We'll put a little one here. Okay, and these would be held together at the centromere. So that would be our paternal chromosome. This would be our maternal chromosome. That guy's not really at the metaphase plate. Let's put him at the metaphase plate. Okay, you're going to have something akin to this, all right? What's going to happen when those 2 cells divide, okay, is our metaphase plate's going to run this way, just so we put that in, okay? You're going to move the homologous pairs over to this side, over to this side, over this way, okay? What you would generate over here would be a cell that has strictly maternal or strictly paternal. Oops, that one's supposed to be the little one. I'm just making all kinds of mistakes today, all right? Whereas over here, if we bring these to each pole, all right, what we'd end up with in this cell is the large paternal chromosome and the little maternal chromosome, whereas this cell would have the large maternal chromosome and the little paternal chromosome, okay? And I have a diagram that shows this in the PowerPoints as well. But the independent assortment really gets at how those homologous pairs line up at the metaphase plate, okay? Now, with just putting in 2 chromosomes, there's not a whole lot of options that you've got there. But when you think about this in terms of the human genome and having 23 pairs, now you've got a lot more diversity as to whether or not you've got a maternal chromosome 1, chromosome 2, 3, 4, etc., all the way down to chromosome 23 and the X, or 22 and the X and the Y's. Okay, so that's what we're talking about with independent assortment. We're talking about how the homologous pairs line up at metaphase. Okay, and this can be either metaphase 1 or metaphase 2. Okay, you can get variation at either one. All right, I've drawn it at metaphase 1. It would also be appropriate to put it at metaphase 2 also, because when those chromosomes line up on the metaphase plate again, there's going to be some assortment as to what goes where. All right, and that'll increase some variation. And with metaphase 2, the part that's going to play a little bit bigger role in the independent assortment at metaphase 2 is going to be our third type of variation. Okay, and that third type of variation is called crossing over. Okay, and we're not talking about, in this case, going to the great beyond. We're talking about crossing over in genetic terms. We're talking about essentially trading or exchanging genetic material. And this is going to have a particular phase of meiosis that this will occur. Crossing over is going to happen during prophase 1. And I'm going to draw that again over here, okay, because this is where I have space. So we have our cell. We're in prophase 1 of the first meiotic division, okay? What's going to happen, okay? And I'm going to draw this first here, and then I'll put it in our cell here, okay? What we're going to have happen, right? We've got chromosome number 1 from mom, and we've got chromosome number 1 from dad, okay? Now what's going to happen, excuse me, during prophase 1, okay? And I'm going to draw these next to each other just for simplicity's sake. Okay, so I'm going to go with some straight lines now to show you how this works instead of my typical X's. All right, is those 2 chromosomes come in such close proximity that what they do, I'm going to draw this in as orange, is they synapse, okay? Or they touch each other, essentially is what's going to happen. All right, there are going to be proteins that are going to hold those chromosomes together. And then what can happen is that you'll get a break, a physical break in the chromosomes. And what you'll have essentially is, oh, I didn't draw that right. Let me start that again. All right. This is why I don't ask you to draw this stuff on the on tests, okay? So as long as you know the concept, I'm going to be happy, okay? So what you're going to have happen, all right, this is a little better drawing of that, okay, is those chromosomes are going to kind of physically touch, and what happens is there can be breaks that are induced into that chromosome. And then what happens is you end up generating some hybrid chromosomes that are completely unique, okay? Because what we have here then, we've got blue, that one's all blue, and then we've got to add a little bit of pink here, okay? Is you're going to generate some new combinations, okay? So this is going to be the crossing over event, okay? What you're going to have where you've got those bits crossing over, and I'm going to draw that in green, is going to be a chiasma, all right? That's where those homologous chromosomes will interact. And what happens in those homologous regions, there can be small breaks that happen. And essentially what you get is a trading of the genetic material. What was part of the paternal chromosome attaches on to the maternal, maternal attaches on to the paternal, okay? So these two in the middle, they're still chromosome 1, but they're now considered recombinant because it's not like the original, okay? So I've left the maternal, original maternal here, original paternal here for you to see, okay? And I've drawn this as just one crossing over. You can actually have multiple areas where you have crossing over occur on a chromosome. It all kind of depends on size. The bigger chromosomes, like chromosomes 1 through 5, you're going to have lots and lots of areas where you can have crossing over. The smaller chromosomes, like chromosome 20 to 22, you might get one, you might not get crossing over because they're smaller. You've got less genetic material there, okay? So the size of the chromosomes, what's going to pretty much dictate how much crossing over you've got that occurs. I've just drawn it as one. I think I've got a diagram in your PowerPoints that shows multiple crossing over. But that's essentially how you can generate additional variation, all right, is by crossing over during prophase 1, okay? So what we'd have happen then in our cell is you would have, oh, my pink's running out of ink. There we go, okay? With our chromosomes, they're going to exchange information, okay? And I'll try and draw a longer one in here and see if we can't do this multiple times. Depends on my skill level here, okay? But you could also have, you know, something like this and end up with multiple crossovers that happen and multiple chiasmas for the chromosome, okay? The key on this is that process of crossing over has to involve the homologous pair, okay? So you'll get crossover between chromosome 1, that's maternal and paternal. You'll get crossing over that will happen between chromosome 2, maternal and paternal. But you won't, for example, get material from chromosome 1 crossing over with chromosome 2 that's paternal, okay? Now, in terms of that, you do get some recombinations like that that will happen. Those tend to be more detrimental, though. Those tend to involve mutations, and I'll talk a little bit about that more when we get to genetics specifically. But for this unit, okay, what I want you to concentrate on is what happens normally. I'm not asking you for mutations. I'm asking you normally. Normally, crossing over is going to happen between the homologous pairs, okay? And those homologous pairs, you will also often see those referred to in textbooks as tetrads, okay? I haven't used that terminology with you yet, but that is the correct terminology to describe what's happening during meiosis 1. You've got the tetrads. You're talking about the homologous pairs that are lining up and matching up during the process of meiosis 1, okay? So those are going to be tetrads, all right? Hopefully that makes sense. If you have any questions,